Usually when we talk about getting a medical diagnosis, there's this expectation of precision, right?
No, yeah, like engineering.
Right. Like you break your arm, the x-ray shows that jagged white line, and the doctor just points at it and says, there it is.
It's totally binary. It's broken or it's not broken. It's visible, it's categorized, and, well, honestly, it's comforting for patients to have that clarity.
Exactly.
Yeah.
But then you step into the world of chronic diseases, specifically diabetes, and how it damages the body over time, and suddenly that x-ray machine is just useless.
Completely useless, yeah.
We find ourselves looking at a diagnostic landscape that is just incredibly murky, especially when we talk about diabetic nephropathy, diabetic kidney disease.
Right.
And the statistics for this are just staggering. We are talking about a condition that eventually affects roughly 30% of people with type 1 diabetes and up to 40% of those with type 2 diabetes.
Yeah, the sheer scale makes it just a massive global health burden. And I think the most frustrating part for both patients and clinicians is that our current therapeutic options act as little more than speed bumps.
Speed bumps, wow.
Yeah, I mean, they attenuate the disease progression, meaning they slow the damage down a bit, but they completely fail to halt its onset or its advancement. It has always been this one-way street toward kidney failure.
Until now, maybe. Because today, our mission on this deep dive is to explore a truly groundbreaking 2026 study published in Cell Death and Disease by Hewis and colleagues.
It's a fantastic paper.
It really is. This paper completely redefines how diabetes actually destroys the kidneys. It shifts the blame away from the blood vessels and points the finger directly at a specific writer enzyme that is prematurely aging your kidney cells.
Which is a huge paradigm shift.
Right. So if you've ever wondered why high blood sugar causes so much systemic damage, you're going to want to hear this. Okay, let's unpack this. What if the secret to stopping kidney failure isn't in fixing the physical filters, but in stopping the cells themselves from turning into what this paper essentially describes as zombie cells?
Yeah, that is the core paradigm shift of this research. And at the center of this shift is something incredibly small, but, immensely powerful. We're looking at an epigenetic RNA modification called N4-acetylcytidine.
That's a mouthful.
It is, yeah. We can just call it ac4C for short.
Okay, ac4C.
Right. And if you want a mental image, think of an ac4C modification as a chemical Post-it note, basically attached to a cell's internal instruction.
Oh, I like that.
Yeah, and in the context of this highly stressed diabetic environment, that Post-it note is essentially screaming at the body to accelerate cellular aging.
But before we get to the microscopic chemical tags and Post-it notes, I think we first need to understand, where the damage is happening and what that damage actually looks like on a tissue level.
Absolutely.
Because this paper fundamentally changes the map of the kidney for us. Think of the kidney tubules like a city's highly efficient water recycling pipes.
That's a good analogy, because historically, diabetic nephropathy has been viewed almost strictly as a glomerular disorder.
The glomerulus.
Right. The glomerulus is the kidney's initial filtering unit. It's the sieve that separates the waste from the blood.
Which makes sense intuitively, right? Like if the filter breaks, you get proteins leaking into the urine, which is the classic clinical sign of kidney disease.
Exactly.
But this paper proves we've been ignoring a massive piece of the puzzle. It highlights that renal tubular injury is equally pivotal.
Right.
After the glomerulus does the initial filtering, the renal tubules are the pathways that do all the heavy lifting, reabsorbing all the necessary nutrients and water back into your blood.
And what the researchers found is that the epithelial cells lining these tubules are undergoing a dramatic, totally toxic transformation.
The paper refers to it as renal tubular epithelial cell senescence, which sounds like a fancy academic way of saying the cells are just getting old. But it's much more destructive than normal aging, isn't it?
Oh, far more destructive. It's a highly specific, totally dysfunctional state. The researchers modeled this in the lab by exposing these tubular cells to severe stress, specifically 30 millimolar glucose concentrations.
Wow. Which is simulating like aggressive, uncontrolled diabetes.
Exactly. And under that extreme high sugar stress, these cells enter a state of permanent arrest. They stop dividing. They stop doing their normal jobs of reabsorbing nutrients.
Right.
But crucially, they refuse to die. They just sit there secreting these toxic inflammatory signals into the surrounding tissue. They literally become zombie cells.
So going back to that water recycling pipe analogy,
Yeah.
Senescence is like those pipes rusting out and the maintenance workers, the cells just sitting down on the job.
Yeah, they go on strike.
Right. They refuse to work, but they also refuse to clock out and leave. They just take up space, tossing their trash everywhere, which eventually causes a cascading system failure across the entire kidney network.
That is a highly accurate way to visualize it, and we can actually measure that trash they're tossing out. The researchers tracked very specific biological red flags or markers of this senescence.
What kind of markers?
Well, when these cells turn into zombies, they dramatically crank up the expression of proteins like P16, P21, and P53.
Okay, and in cellular biology, those three proteins act as the emergency brakes on the cell cycle, right?
Yeah.
They permanently halt growth.
Exactly. They pull the e-brake.
The researchers also looked for a specific enzyme called SA-beta-gal, didn't they?
Yes, they did.
Because from what I understand, they can literally drop a chemical stain onto the lab dishes, and any senescent cell pumping out this enzyme turns a deep, bright blue.
Yeah, the senescence-associated beta-galactosidase stain. It gives a really clear visual confirmation. But they went even deeper into the damage. They looked at a marker called gamma H2AX.
Okay.
Specifically, they looked at phosphorylation at serine 139.
Okay, let's decode that a bit for the listener. What is actually happening at serine 139?
Right, so when a cell's DNA physically breaks, specifically a double-strand break, which is catastrophic damage, the cell sounds an alarm. It attaches a chemical tag at that exact spot, serine 139, on a structural protein to call for repair.
Oh, wow.
Yeah, so massive spikes in gamma H2AX mean the cell's genetic code is literally being shredded by the glucose stress.
That's terrifying.
It is. And to prove the tubules themselves were structurally failing in response to all this, they tracked massive spikes in two specific tubular injury markers known as KIM1 and NGAL.
Which are essentially structural SOS signals.
Exactly.
So if we pause and look at what we've got so far, diabetic kidney disease isn't just a failing glomerulus filter. The renal tubules, the reclamation pipes, are equally responsible.
Yeah.
And the high glucose levels are forcing these tubular cells into a toxic, arrested zombie state filled with DNA damage.
Right.
It makes you wonder, looking at your own daily habits when you consume excess sugar, how might your own cells be forced into this accelerated aging response just to survive the metabolic stress?
It's a sobering thought.
It really is. But sugar alone isn't a blueprint. It's just fuel, or in this case, stress. What is actually flipping the switch to turn these specific cells into zombies?
Well, to find out what flips the zombie switch, the researchers couldn't just guess. They looked at actual human kidney biopsies, and they analyzed single-cell transcriptomic data from early-stage human diabetic nephropathy.
Right, using a database, right?
Yeah, they use a public repository called the Kidney Interactive Transcriptomics, or KIT database.
The KIT database.
Right, and by looking cell by cell at which genes were turned on or off, all the data pointed to a massive overactivity of one very specific enzyme.
And acetyltransferase 10.
NE10.
NE10 is the absolute linchpin here. It is currently the only known writer enzyme in human biology that can apply that ac4C chemical modification we mentioned earlier directly onto RNA.
So if mRNA is a text message sent from the cell's nucleus carrying instructions on what proteins to build, NE10 is the molecular pen writing the post-it notes on those messages. That's a great way to think about it. And when they looked at the human biopsy data, NE10 was massively elevated, specifically in the renal tubules of patients with diabetic kidney disease.
And the clinical correlations they found were wild. Like, instead of just a list of markers, think of a patient's lab results completely crashing. The researchers saw a perfect, tragic mirror effect.
Oh, it was a 1-to-1 correlation.
Right. As the levels of NE10 in the kidneys went up, all the good numbers, the eGFR, which is your kidney's overall filtration rate, your hemoglobin, your blood albumin, those all tanked.
Conversely, the waste products building up in the blood skyrocketed.
Right.
High NET10 meant dangerously high blood urea nitrogen, high serum creatinine, high beta-2 microglobulin, high cystatin C, high uric acid, and high total cholesterol.
It's basically a full systemic failure.
Yeah, it correlated perfectly with the kidney's decline.
It's like NET10 is the conductor of a very toxic orchestra, and it perfectly matched the rise of that aging marker, P16, that you mentioned earlier.
And what's fascinating here is that they didn't just see this in human observational data. They saw the exact same spike in NET10 and a corresponding massive increase in the ac4C tags in two highly controlled mouse models.
To make sure it wasn't a fluke.
Exactly. They used STZ-induced mice, which models type 1 diabetes, and spontaneous DBDB mice, which models type 2 diabetes.
Okay.
In both distinct forms of the disease, the NAT10 enzyme was in absolute overdrive.
Let's expand on that text message metaphor to visualize what Net10 is actually doing. Because if mRNA is the text message with instructions, Net10 isn't just any pen.
Or it's not.
It's a molecular highlighter pen. By adding the ac4C tag, it's basically highlighting the text message in neon yellow ink. It makes the message highly stable.
Yes.
So instead of the cell reading the message once and then deleting it, the cell reads it over and over and over again, building massive amounts of whatever protein that message codes for. The ac4C tag enhances translation efficiency and mRNA stability.
It literally forces the cell to mass produce whatever instruction is highlighted.
So we know Net10 is an enzyme acting as a writer, adding ac4C tags to stabilize RNA. And we know it goes into overdrive in diabetic kidneys, perfectly matching the decline in function.
Right.
Which leads to the obvious question. If a single highlighter enzyme perfectly triggers kidney failure, what specific message is it highlighting? What is the instruction that Net10 is forcing the cell to read on a loop?
Well, to discover the highlighted message, the researchers had to use an incredibly advanced sequencing technique called ac4C on the diabetic mice.
ac4C, okay.
Yeah, this technique allows them to fish out and read only the specific pieces of RNA that have the ac4C tag physically attached to them.
Oh, wow. So they isolated the highlighted messages.
Yeah.
And what did the sequencing reveal?
They found 162 differential ac4C peaks.
Meaning what exactly?
This means there were 162 specific spots where the highlighting was significantly different in diabetic kidneys compared to healthy ones.
Okay, so a lot of noise.
A lot of noise, yeah. But by cross-referencing this with overall gene expression data, one single candidate stood out far above the rest, which was PAPP-A.
Pregnancy-associated plasma protein A, which is a wild name to pop up in a study about diabetic kidney failure.
It is surprising, yeah. It was originally discovered in pregnant women, where it plays a role in fetal development, but fundamentally it's a metalloproteinase that regulates cell growth factors in tissues.
Okay.
And the sequencing data was undeniable here. The rogue NAT10 enzyme was directly binding to the PAPP-A mRNA and plastering it with ac4C tags.
But, in biology, just seeing a tag on a piece of RNA doesn't prove it's doing anything. Like maybe the tag is just a molecular decoration. How do they prove the highlighter actually stabilizes the message?
That's a great point. To prove causality, they used an actinomycin D chase assay.
Okay, actinomycin D. So to prove it stabilizes the RNA, you basically need to stop the cell from making any new RNA and just watch how long the existing stuff survives, right?
You've just described exactly how the assay works.
Oh, nice.
Yeah, actinomycin D is a compound that completely halts all new RNA synthesis in a cell. It stops the printing presses.
Okay.
Any mRNA left in the cell will naturally start to degrade over time. By using this biological freeze frame, they could measure exactly how long the PAPP-A mRNA naturally survives.
It's like a molecular countdown timer.
Yes, exactly. And they found that when they silenced the NATA10 enzyme, the PAPP-A mRNA degraded rapidly. It just fell apart.
Huh.
But when NATA10 was active and adding those ac4C tags, the PAPP-A mRNA became incredibly stable. It survived significantly longer, meaning the cell kept using it to churn out PAPP-A protein.
Okay, here's where it gets really interesting. PAPP-A isn't just a bystander floating around in the fluid. The researchers used a cutting-edge artificial intelligence tool called AlphaFold3 to predict the physical shapes of these proteins, to see who PAPP-A might be interacting with.
And AlphaFold3 provided a vital clue. It predicted, with a PTM score of -0.61, that PAPP-A physically interacts with P53.
Okay, a PTM score. What does that mean for us?
A PTM score measures the confidence of the protein folding structure. So -0.61 strongly indicates a highly probable physical fit, like a key sliding perfectly into a lock.
And P53 is one of the most famous proteins in biology. It's the master regulator, the tumor suppressor. It's the protein that forces cells into aging and arrest when they are stressed.
It is the main conductor of the senescence signaling pathway.
Right.
And the researchers didn't just trust the AI's prediction, they proved this physical lock and key interaction in the lab using co-immunoprecipitation, or co-IP, and his pull-down assays.
Which are what exactly?
These are techniques where they essentially use molecular handcuffs to physically pull the proteins out of the cell together, proving they were physically locked together in vivo.
Okay, let me make sure I'm holding all these pieces together in this crazy Rube Goldberg machine.
Let's hear it.
Step 1. The severe stress of high glucose triggers the NET10 enzyme. Step 2. NET10 takes out its highlighter pen and adds ac4C tags specifically to PAPP-A mRNA. Step 3. Because of the tags, the PAPP-A mRNA becomes ultra-stable, and the cell creates tons of PAPP-A protein. Step 4. all this excess PPPA physically binds with the master regulator, P53.
Spot on.
Step 5. This physical interaction hyperactivates P53, which then turns on the aging pathways, cranking up those cell cycle breaks we talked about, P21 and P16. And finally, step 6. The cell permanently arrests, becomes a zombie, and starts destroying the kidney tubule.
That cascade is the exact molecular engine driving diabetic tubular senescence.
Okay, but hold on. PPA is supposed to be associated with pregnancy, creating life, regulating growth factors. Yeah. How do we know it's the actual executioner here and not just a protein that happens to be in the wrong place at the wrong time?
That's a fair question.
Like, we know NAT10 specifically targets the mRNA for PPA, and we know PPA physically binds with P53 to trigger aging. But is PPA the true cause? It's fascinating to consider how a protein vital for one stage of life could be hijacked by a disease to accelerate aging in another part of the body entirely. Biology is incredibly resourceful, for better or worse.
It really is. But to answer your skepticism about whether PAPP-A is the true executioner, the researchers had to move from theory to therapy.
Okay.
They went back to the human kidney proximal tubular cells, the HK2 cells, in the lab. They flooded them with the 30 millimolar high glucose stress.
The 30 millimolar stress, right?
Yeah. But this time, they used short hairpin RNA, or shRNA, to genetically silence the NAT10 enzyme.
Let me guess, without the highlighter pin, the cell couldn't read the aging instructions, so it stayed healthy.
Yes. The cells didn't turn into zombies. There was significantly less of the blue SA beta-gal staining, less P16, less P53, and less of the DNA damage alarm marker gamma H2AX.
So removing NAT10 completely saves the cell.
It does. But here's the definitive proof you asked for regarding PPA.
Okay.
In those same saved cells where NAT10 was silenced, the researchers artificially overexpressed PAPP-A. They forced the cells to manufacture the PAPP-A protein anyway.
Oh, wow. And what happened?
The moment they did that, the protective effect vanished. The cells instantly turned into zombies again, despite NAT10 being completely gone.
Wow. So PPA is absolutely the bullet that kills the cell. NAT10 is just pulling the trigger.
Exactly. Now, proving this in a petri dish is one thing. Understanding the mechanism is great for textbooks, but to mean anything for a living, breathing patient, they had to prove this works in live animals.
And they went all out for this, didn't they? They didn't just use standard mice.
No, they didn't.
For the type 1 diabetes model, the STZ model, they created a custom-built conditional knockout mouse. The genetic designation is CDH16-Carina10 flox.
Which is a mouthful.
It is a mouthful.
Yeah.
But what it essentially means is that they engineered a highly specific mouse where they could inject a drug tamoxifen that acted like a genetic scalpel. It functioned as an ignition key to delete the NAT10 gene exclusively in the cells that have the CDH16 promoter.
Right.
And those happen to be only the kidney tubules. The rest of the body was completely untouched.
It's an incredibly elegant genetic scalpel. And the results were undeniable. When they induced type 1 diabetes in these tubule-specific knockout mice, there was a massive drop in the urine albumin to creatinine ratio.
Which is the gold standard for kidney health.
Exactly. The kidney injury was fundamentally halted. The tubules didn't age.
And they took a completely different approach for the type 2 diabetes model. They used an adeno-associated virus, or an AAV. So they basically used a viral delivery truck carrying the instructions to silence NAT10.
Yeah.
And they injected it directly into the mice.
Again, the AAV acted as a targeted genetic therapy. It rescued the broken brush borders in the kidney tubules. Those are the microscopic hairs that absorb nutrients.
Right.
And it stopped the expression of the aging markers P16 and P21.
Okay, but let's be real for a second. We can't easily do complex viral gene therapy or tamoxifen-induced conditional knockouts on the hundreds of millions of diabetic patients globally.
No, it can't.
It's just not feasible yet. We need a pill. Is there a pharmacological way to actually do this?
Well, if we connect this to the bigger picture, the clinical picture there is, the researchers utilized a small molecule drug called remodelin.
Remodelin.
Right. Remodelin is a known chemical inhibitor of NAT10. They gave it to the diabetic mice intraperitoneally, meaning injected into the body cavity at a dose of 5 milligrams per kilogram.
So they just gave them the drug to shut down the enzyme.
And it worked flawlessly. Systemic administration of remodelin successfully blocked NAT10 activity in the kidneys. It dramatically reduced the levels of PPPA and P53.
Wow.
Yeah, it cleared out the senescent zombie cells, repaired the tubule injury, and fundamentally protected kidney function in the live diabetic mice.
So what does this all mean? It means we actually have a druggable target. We aren't just looking at the rusting pipes and shrugging our shoulders anymore. We have the chemical wrench to stop the rust.
It represents a monumental shift from just managing the symptoms of diabetic nephropathy, like just trying to lower blood pressure or manage blood sugar, to actively stopping the cellular destruction at its epigenetic root.
Let's pull all of this together. We know genetically removing the NET10 enzyme strictly in the kidney tubules completely prevents the cellular damage caused by diabetes.
Yeah.
And we know a small molecule drug called remodeling mimics this genetic knockout, offering a highly realistic pharmacological treatment for diabetic kidney disease. If we can use a drug like remodeling to literally turn off the aging switch in diabetic kidneys, what other aging processes in your body might be controlled by this exact same mechanism?
That is the true frontier here. I mean, this study forces us to look beyond traditional clinical targets. We spent decades focusing on the glomerulus filter. But by following the transcriptomic data down to the single-cell level, these researchers found the answer hidden in RNA epigenetics within the tubules.
It's amazing.
It fundamentally changes how we approach cellular senescence across the board.
What a journey. We went from the macro systemic problem of diabetic kidney failure, zoomed all the way into the microscopic tubules to find those zombie cells.
We did.
We discovered the NAT10 writer enzyme holding a neon highlighter pen, saw how it uses ac4C tags to stabilize the PAPP-A message, watched PAPP-A act like a lock and key on p53 to trigger aging, and finally learned how a drug like remodeling could halt that entire cascade in its tracks.
It really is a masterclass in molecular detective work.
I want to leave you with a final thought to mull over today. We learned that NAT10 adds a chemical tag that tells kidney cells to age prematurely when they're exposed to high sugar. But ac4C is a fundamental RNA modification found throughout human biology.
Right, everywhere.
It's not just in the kidneys. If RNA writers like NAT10 are holding the pen that writes our biological clock, could targeting these epigenetic enzymes eventually lead to a universal eraser for human aging itself? Think about that the next time you look at a spoonful of sugar. Thank you so much for joining us on this deep dive. Stay curious.